Thank you, and I'd like to invite the last speaker in this session from the Amazonian isomer, Joe Vinetz from UCSD.
Thank you very much. I am what keeps you from lunch, and I understand that. I also want to recognize the contributions of the historical popes to the difference between the non-Amazonian and the Amazonian isomer, who, remember, the Pope in the 16th century split South America into Spanish-speaking and Portuguese-speaking. So we're the Amazonian isomer, and we do both. We're ecumenical. I also wanted to thank Dr. Agre and Dean Clegg, Dr. Casadevall, for really organizing this fantastic salute to a World Malaria Day. Wow, what a diverse set of speakers and ideas, all malaria all the time, and it's really quite mind-expanding for me. And it's really nice to be back here at Hopkins because I spent a lot of formative years here. So without further ado, I want to, at the outset, recognize some of my collaborators. Almost all of this work is done in Peru and Brazil quite independently. This is at the TROP-Med meeting last year with our fearless leader, Mala. I also wanted to, and during my presentation, I'm going to be acknowledging various contributors at the time. I also want to point out the rock star of South American malaria, Dionisia Gamboa, who twice has won the L'Oreal Women in Science Award. She's really based on her malaria research for the last decade. 10 years ago, there was no academic malaria research in Peru, period. And now, well, you can judge what we've accomplished, but what we've done in Peru really is very much led by her. So without further ado, I'm going to move forward. Okay. All right. So the agenda today is I'm going to talk about the unifying theme of the Amazonia isomer. I'll get to that in a moment. And this is really, I think, a generalizable and global identification of barriers to control and elimination throughout the malaria endemic world. I'm going to talk about epidemiology that contrasts and has certain similarities between Brazil and Peru. In Brazil, where there's disappearing residual as well as emerging malaria, our study sites in Brazil and Peru are actually quite close to each other within a few hundred kilometers. The only malaria in Brazil is in Amazonia, and we're focused on the highest spots in western Amazonia in Brazil. And in Peru, we've had persistent, but in the last 3 years, alarmingly increased hypoendemicity, and I'll talk about that. I'm going to mix that together with the Amazonian malaria vector, Anopheles darlingi, in terms of ecology, how anthropogenic influences has changed the behavior and the genetic differentiation of that mosquito species into, we can't really call it incipient species, but there's some big stuff which really reflects what's going on in Africa as well, driven by human, not just deforestation, but by bed net use, probably by indoor insecticide use, to the point where we see outside transmission rather than inside transmission, which is the so-called residual malaria. And finally, I'm going to talk a little bit at the very end about how we use this platform to enable studies really of vivax sporozoite biology, one of the great gaps in the whole malaria field. So that's the agenda. Our broad long-term goal is to actually, it's very modest. It's just to eliminate malaria in Amazonia. This is a low transmission setting, and what Socrates pointed out is a truism we figured out more than 10 years ago. You don't need many infections to become clinically immune. You don't need many infections to become asymptomatically parasitemic. Why is that? And that really, and for more than a decade, we have been asked by the Africa-oriented malariologists, how can this be? And the fact is, it is. And that's something that we're going to talk a little bit about and explore more in the future. The other thing I want to point out is that our tactics for malaria elimination are going to be identifying key gaps in the field. And we try to identify local as well as generalizable gaps. We have to communicate our findings, and we do so. This right here, this is just from about a month ago in Iquitos, Peru. I would say 5 years ago, the government ministries just weren't interested in us. And now there's alarming increases in malaria, especially falciparum in the region. And this is in a room at a local hotel, but there was no room to be had because this is alarm levels in Iquitos, Peru. So this is one of a number of regular communications with ministries of health that we have every few months, really for the last several years. And this is just another example of that. Let's go next. Okay. So our themes are of our isomer is that asymptomatic subparasitemia drives hypoendemicity. That's just a few words that communicates a very powerful concept. And the idea is twofold. And first of all, if you're asymptomatic, and we've had a number of debates among the isomer group, what is actually asymptomatic? And for me, it's somebody that wants to go play soccer, okay? So it's pretty simple, and we want biomarkers and we want objectivity, but people that are going through their activities of daily living, that's what we think of as asymptomatic, and we query people about the past 2 weeks. Do you have anything, any fever, you have headache, do you feel like working, do you think you have malaria, et cetera? And it turns out, and I'll show you some data, that's actually quite predictive and heuristic for actual clinical immunity or so-called non-sterilizing immunity. Second, and I've had problems for years trying to figure out how to communicate this, but the idea is that we have silent, mobile human reservoirs of transmission. Finally, people have helped me with the wording. The idea that people move around with silent potential for transmission, that's what malaria elimination is all about. And you've heard a number of asymptomatic parasitemia issues come up today. It's that silent mobile reservoir. That's kind of like my Broadway show tune here. That's what you should whistle when you walk out the door today, okay? A Broadway show is no good unless you whistle the tune on your way out, okay? Another issue that's come up is actually how human activities basically lead to changing mosquito behaviors, and that's actually led to residual malaria. And in Peru, we've actually got a one-year TDR grant just to look at residual malaria to contrast with what's going on in Africa. So the third theme is we enable local researchers. Here's Dione Gamboa, Alejandro Llanos, who are the Lima-based collaborators, with Hugo Rodriguez, who was the Ministry of Health Director in Iquitos and now is a collaborator of ours. These are highly political positions, and people last weeks to maybe months, sometimes a year. But this gentleman is from a very rural, distant place in the Amazon and has done more to connect the science to the policy than anybody that I've seen since 1998. So this is the idea of enabling local researchers and also students and other trainees for capacity building. Okay, so I'm going to talk about Brazil, Peru, in terms of epidemiology. We'll talk about ecology of vectors, a little bit of transmission biology, a little Vivax sporozoite, and we'll be done. So Marcelo Ferreira, who's a professor of parasitology at the University of Sao Paulo, leads this project, which he's called Epidemiology of Disappearing Malaria in Rural Amazonia. He's a remarkable PI because he actually goes out quarterly with his team and takes blood smears himself. And I challenge any of the other project leaders to be doing the same. He's really good, and he's just a lovely guy. So malaria in Brazil now has about 150,000 cases a year. There's almost 30 million people at risk. It's all in Amazonia, 100% of it. And out of those 147,000 cases, there's maybe 3 dozen deaths a year. Not so many. It's all in the Amazon. And Vivax accounts for the vast burden of malarial disease, but that's actually a changing target. One thing that changes, it's different between Brazil or Amazonia and African malaria, is that it's much more equally distributed across age groups. And that implies transmission patterns that are not necessarily house-based, but also related to occupation, and again, that silent reservoir of mobility. So there's a real difference. You don't see that very young or the 5 to 8-year-old age group affected so much. It's really quite all age groups. Okay, so Since the since 1960, so DDT was really the dominant way of controlling malaria in the Amazon, which is reflected in these low numbers of Vivax and falciparum, and we'll see that in Peru in a moment. And then around this time here is when DDT use was stopped. Actually, it was probably slowing down. It was absolutely stopped around this time point. And Brazil had in the late 80s, had a big peak of malaria epidemics. And so you saw the big peaks of falciparum, and between falciparum and then the rest is Vivax. New forms of drug resistance came in. Death rates went down with using better drugs. But nonetheless, the trends of epidemiological trends have really consistently been lower and lower and lower malaria ever since until the current times. Right now, malaria is now fragmented. It kind of goes along with forest fragmentation in a sense. It's a fragmented microgeography, very heterogeneous, as you can see, distributed. This is the falciparum hotspots, and Vivax, just multiply the falciparum cases by 4 to 8, and you'll find Vivax in the same places. So this is in, again, the western Brazilian Amazon, called the Joá Valley. There's a number of places within this area, but the take-home message is this microheterogeneity and fragmentation of transmission. It's very hard to get to. If you think of a lung, you think of a trachea going into the bronchi, and you're getting down to the alveoli, all right? This is the very far away rural areas where it's really hard to get access to. And the surface area is so much bigger than those main areas where you have easy access to. So this is the problem throughout the continent in malaria. It's this microheterogeneity of these hotspots. We're going to talk now about a small settlement called Remencinho that Marcelo and his group worked on from 2010 to 2014, beyond the time when malaria was actually eliminated from the area. So here is Amazonia. Peru's over here. This is Brazil over here. And then you've got this is the Joá Valley here. And you can see that we go down to this really tiny little study site. And each of these dots over here on the right is an individual house done by remote sensing. It's a farming settlement that was opened in 2007, so just 9 years ago, about 250 inhabitants, fairly young, people that came into the area from non-immune, so these are considered susceptibles. He was able to, with quarterly active PCR-based surveillance, it was hard to do microscopy, so this is all molecular. You can see that, first of all, looking at the right, falciparum was essentially gone during this surveillance over about 18 months. And then during those continued surveillance, it was just simply gone. Falciparum in South America is easy, relatively easy to get rid of. And again, it's all about the biology of that hypnozoite. Because you can see here with Plasmodium vivax, prevalence of parasitemia went down in this small community, yet it remains somewhat persistent. It's still there, presumably by reintroduction through relapse. And we know that relapse is usually asymptomatic. So this is part of the malaria problem in low transmission settings, especially with Vivax, because you'll get continued reintroductions over time. Okay. So this is a summary slide. This is published in PLOS Neglected Tropical Diseases a couple years ago. Out of all the Plasmodium vivax infections, 50% of them are subpatent. So when we do microscopy, sometimes they're patent, sometimes they're not. The majority are asymptomatic. Everybody has gametocytes using RT-PCR based on PVS-25, period. And so we don't know what that means, and we haven't done the sexual differentiation looking at males and females, but we know that everybody's got gametocytes. What has not been done, so this is all in venipuncture blood or in finger sticks, but we really don't know what's going on in the skin. We assume that the finger sticks represent what's going on in the skin, and that has to do with direct feeds with mosquitoes on infected people. I'll get to that a little bit later. But the idea is that asymptomatic parasitemia is the rule, not the exception, and it happens in hypoendemic regions. Okay. So these are the folks on Marcelo's team working in this area, and I'm just going to move on to Peru. Again, we thank the Pope for giving the western part of the continent to the Spanish-speaking people. Okay, so there's going to be two stories from Peru, one from longitudinal cohorts in riverine and highway settings, and I'll get to the remote setting in a moment, led by Dione Gambon, Alejandro Llanos, Angel Rosas, and others, a remarkable team that was not together 10 years ago and now operates really quite independently. This is from Dioni's lab. This is at Cayetano Heredia in Lima. Okay, so the history of malaria in Peru from 1940 to the present, from the times of World War II, is that, again, there was some malaria. We don't know how good these numbers are, but we know it's nearly eliminated, quite well controlled through the early 60s, mid-70s, and then DDT stopped again. And then in the mid-1990s, a few years after Brazil, there was this massive explosion of both falciparum and vivax, including a lot of deaths that happened due to falciparum. Chloroquine resistance was the rule, and so mefloquine had to be introduced. Government policy had to be changed. And so control measures have led through a number of ways to relatively okay control until around 2012 or so when it started rising again. This arrow here is important because the Global Fund for HIV, TB, and malaria funded some operational research and implementation science in Peru. It seemed to be fairly effective to help the incidence rates of acute malaria go down. However, there was also a whole bunch of weather-related events. So there's a number of issues that were going on in the Amazon. And then this rise here was heralded by massive flooding in 2013. Amazonian river water height turns out to be the main driver of vector breeding and malaria rather than rain, because it rains kind of all the time. It either rains more or less in the rainforest. But it's really the river height that determines the mosquito biology. And then the other thing, which I'll talk about, I think, in a moment, is about the fish ponds and other anthropogenic changes, because the vector Anopheles darlingi has changed from being a forest mosquito living and breeding in rivers with nice clean water, and now we'll go to fairly murky water. It's a very plastic mosquito with anthropogenically driven differentiation. Anyway, all of these things go on in this part of this graph, and to zoom in on that, this is what's been going on since 2012. Top here is so this is northeastern Peru, the department of Loreto where we work, and Iquitos is down over here. But what you can see here is vivax, and this graph is falciparum, and you can see from around 2,000 cases of falciparum in 2012, there's more than 12,000 cases in 2015. So that's why that meeting room was so crowded, because it's an alarm. They have criteria, and people are seriously concerned at the ministry level. And then they asked the isomer, because we had developed relationships over several years, they said, can you please come and help us? Okay. So this is exactly our view of what one of the most important contributions we can make, which is communicating the science. The science is going to underlie policy, and that's why this graph, I wanted to go through this graph. Okay, so our study designed in two communities. They're called Cauide and Lapuna. Cauide is down here. It's on this road between Quito City, which is up here, and it's a road. It's actually a 120-kilometer road. A remarkable thing in the Amazon, which is simply a swamp. In the Peruvian Amazon, this is the road. It's done for commercial reasons more than 15 years ago. So Cauide is down here, and then Lapuna is, Iquitos is over here, but the Lapuna is a riverine setting. And so we've had about 1,000 people under active surveillance with weekly visits for symptoms, monthly blood smear sampling for microscopy and PCR for the last several years. So that's the basic study design. And I'm not going to show a lot of data for the purpose of time, but what I can tell you is that the solid lines here represent PCR, and the dotted lines here represent microscopy. And these are the monthly prevalences from December 2012 to September 2015. And what you can see in the village of Lapuna, this is the riverine setting. It's just essentially there's about a 10 to 25% prevalence of parasitemia in that village by PCR nearly the entire year. Microscopy, which is the government ministry's go-to, there's no rapid diagnostic tests. There's no rapid tests at all that are used by the government now that the Global Fund money ran out. So the microscopy, which is down here, is obviously substantially less. The patterns of seasonality are different between Lapuna, the riverine setting, and Cauide, the highway setting. And we're going to talk a little bit about mosquitoes, which we think it's the mosquito-human interaction that determines these differences in seasonality. One of the important things, and Socrates has similar data, but these are some details. We asked the question, what happens to people when they don't have symptoms and they have parasites in their blood and they don't have a positive smear? It's not clear if you have to treat an asymptomatic positive smear person, but we do anyway, at this point, at least during this. And this is what happened. There were 4 patterns. But the bottom line is 13% of 400 people turned into a thick smear and had a fever over quarterly sampling over a year. 13%. The rest of them didn't. Most of them didn't have fever. Some people, maybe a third, had a positive smear but never had a fever or any other symptoms of malaria. So this is the natural history of, and this is a pretty good sample size. This is the natural history of Vivax infection in a hypoendemic setting where clinical immunity develops. So this is a question that his group and our group have been looking and investigating, and I think this is a pretty good answer. We have some other data associated with it. The infections are essentially single clones. There are very few monoclonal infections because the infection intensity is so low. So from this part of the talk, I would like to suggest that most of the subpatents, subpatents means microscopy negative, PCR positive. So subpatents resolve their infection almost all of the time. And we had debated within the isomer network this simple classification of symptomatology. And it looks like that's heuristically useful by actually talking to people. I'd like to have some biomarkers. We've done a little bit of work. Others have done some work about asymptomatic parasitemia. But just talking to people and finding out if they want to go play soccer is pretty useful. Then we're going to get into this again, reflecting also what Socrates has already presented, is that asymptomatics who develop a positive thick smear, that means they're patent, might actually be the ones who are responsible for transmitting. And we actually have experimental data that I'm going to show you towards the end based upon experimental mosquito infections with Anopheles darlingi. And therefore, about half of the Vivax asymptomatics can potentially transmit. And again, more experiments need to be done to actually do this. This is an experimental question. Because the number that we want to get is out of that vast ocean of asymptomatic parasitemics, how efficiently over time are they able to infect mosquitoes to maintain transmission? What's the integrated area under the curve of malaria parasitemia days in relationship to mosquito exposure? And that's a quantifiable parameter. And that's a really important number that none of the malaria models actually include now. But I think that's critical in this hypoendemic setting. Okay. So the other part of Peru I wanted to tell you a little bit about was Santa Emilia. I've told this story a little bit within the isomer network. So this is another map of Iquitos, Peru. So here's Iquitos. Here's Lupuna, the riverine setting. Masan is another rural river setting. Margaret Kozik and Pablo Peñatoro ran this site for years, which we're back into. They'll be happy to know, my Hopkins friends. Cahuita is down here, and this is this highway. So this is easy to get to malaria. You just get on a boat and you go, or on a road, and in less than an hour you're at the study site. It's fantastic unless there's flooding. Well, here's the issue. If you go over hill and over dale and across a whole bunch of rivers down south, go down this river and go back up this river, you get to this village called Santa Emilia. So there's no Ministry of Health surveillance here. This is 20 hours or so away by travel from Iquitos. It's just too inconvenient for people to get to. And it turns out this is, for us, the poster child of malaria elimination challenges. Okay. So this population has about 213 people. When you repeatedly sample a small village like this in a cross-sectional way, it's essentially a longitudinal cohort study because everybody gets selected with about 80% retention within the sampling scheme. 36 households, fairly young, and we got about 11 or so smears per person over about, well, we're continuing the cohort, but the data I'm showing you here was over about 13 months or so. So between March 2013 and February 2014, we sampled essentially the whole village by blood smear using microscopy. We had a microscope with a mirror on it so we could provide treatment. We brought treatments with us. I can't say we because I didn't go there. But we had a malaria-trained technician who brought medications, and a mirror microscope, and he was making diagnoses. And this guy can catch down to 10 to 15 parasites per microliter of blood. This guy is really remarkable. His name is Daniel Wang. We recruited him back. He left, and he's one of the best microscopists around. And so these are really good data. And he went 13 times to Santa Emilia. When we brought filter papers back to the lab, we had, again, much higher parasitemia by PCR with the dashed lines than with these dotted lines down here. The important thing is that, and I don't have time to show the data, but most of the infections were asymptomatic. Again, what you might expect in this hyperendemic setting. This is a protein array analysis that we did because we didn't actually know when the transmission season was. So in order to figure out a transmission season, you basically use passive surveillance data and you add them up every week and you make a graph. People come to the health post because they have fever, they have a blood smear, and you identify malaria. Well, this was not possible here without research intervention, basically. And so we were able to go back and really ascertain, well, we thought we knew what the transmission season was. And it turns out it was quite different. And the transmission season, again, is determined by, well, we think by the dogma, by rain and by mosquito blooming. And it turns out the mosquitoes there all the time, and I'll show those data in a sec. And we also know that the protein array analysis actually told us. This is unexpected. So we expected that malaria was transmitted in this remote Amazonian village between maybe February, June, July. That was the typical thing that we thought based upon mosquito abundances and passive data. And it turns out it's not the case. So we got two samples from Santa Emilia, one in September and one in March. And we separated them by age less than 15. Again, this is similar numbers in terms of age that Socrates saw, and greater than 15. And that age is really important because that's when kids start working in the field and start working outside the house more. They have good primary school, not such great secondary school in this region. And what you see is that, first of all, there's, and it's ranked by less exposure to more exposure. So you're going to be looking at how much red and gray is going to be at the right part of each of these columns. And so what I'll show you is that there's less, and at the right here is Papua New Guinea. So Papua New Guinea, which is a very high intensity transmission place. And this is sort of the benchmark against which we're comparing the exposure. On the top here, we have falciparum proteins on the array. On the bottom here, we have vivax proteins. So that's the experiment. And we had a fairly decent sample size of more than 150 patients or so. And so what you can see, first of all, is that there's more exposure to malaria in March than there is in September. And that, which means there's more transmission going on between those dates, which is the exact opposite of what we expected. So why is it important to know what the transmission season is? We think, and we have to prove it, and this is our next project period, we think that when you can identify the lowest incidence of disease and the lowest prevalence of infection, that may be the time to start intervening, whether it's at the vectors or with other sorts of either vaccine or drug interventions, which we're not going to do. But you have to know what that transmission season is. The transmission season is it's so simple, and you just look the data. The data don't speak for themselves. We speak for the data, and the data were sitting there. We said, oh, this is really unusual. So we thought this was a novel use of this protein arrays. Okay, so from here, I showed you that the pattern of malaria in the Amazon, in Peruvian Amazon, is different than elsewhere in the absence of Ministry of Health surveillance. That the higher proportion of the villages, of this kind of village, is affected. The vivax dominates, but we also see some falciparum in younger people, which means the parasites being moved around by the silent mobile population. And we used these protein arrays at a genomic scale that led to discovery of new transmission patterns. Okay. So I'm going to end up here. I'm going to go kind of quickly through the vector biology stuff. Anopheles darlingi is the Amazonian vector. It's in a different, it seemed to differentiate from The African mosquitoes maybe hundreds of millions ago. It's in this Nisarhynchus subgenus, and it is the major vector across the entire Amazon region. And if you remember from Socrates' talk, it's not really found in the coastal region. I told you about this Akito-Nata highway. This is the highway region, and we find that that the biting rates are actually higher where there's no forest. The mosquitoes are adapting to where people are. The mosquito, however, is thought to be next to rivers. This is Jan over here, Jan Kahn, who leads the study. And we really don't even know where the breeding sites are. We can't find larvae. And so we don't really know how the larvae and where the eggs are laid are in relationship to human infection. In fact, Jan used, I'm not showing the data, barrier screens that you put, they're about 1 meter high, and you put them close to the ground, and you can figure out which direction the mosquitoes are flying after they bite someone. So that means you can figure out if they're biting inside or outside when they're going to rest after they bite someone. Why is that important? Well, if the mosquitoes aren't staying inside the house and they're not biting inside the house, then indoor residual spraying is not very useful. Okay. And then, and what she found is that the distribution of different types of first of all, the human biting rates was different in the riverine setting versus the highway setting, and we benchmarked this against a previous publication, and we're finding that the riverine setting actually has the highest biting rates for those mosquitoes, and we wanted to ask whether or not that had anything to do with the genetics. I'll get to that in a sec. We find that in these two different sites that the highway biting mosquitoes may bite a little bit earlier, and they have a unimodal biting. This is hours of collection with human biting catches. But the riverine setting actually has two peak times of mosquito biting. So the mosquitoes have differentiated their behavior, and in data I'm not showing you, they've genetically differentiated as well. So this again is environmentally determined mosquito differentiation. And this is just a complicated graph to show that there's differences between peri-urban and chakra. Chakra is kind of, it's not quite the rural areas, and then peri-urban versus forest mosquitoes. And so this has to do with the biting rates, and the biting rates are differentiating, and these mosquitoes have extremely plastic behavior, and therefore, and it turns out it's probably going to be based on genetics. And the genetics, turns out if we just simplify these macrosatellite data into A and B, then a mixture, and these are the different study sites, again, along the river, which is right next to that road, between Coide, the highway site, and La Puna, the riverine site, and the city of Iquitos is over here. We see that there's a different distribution in genetic type A versus genetic type B between the riverine and the highway settings. Which is statistically significant according to chi-square. And using principal component analysis with some dense genomic markers, you can see that there's actually a differentiation of these two mosquito populations along, again, different environmental and ecological settings. Okay, so what I just told you about was that the mosquito, even though it's one species, seems to be going in a bunch of different directions, and we hypothesize that's because it's driven by human behaviors, and complex human behaviors. I'm going to show you just one bit of data about who actually infects the vector. So that's really important, and this is, again, a follow-up. Socrates' lab, which uses Anopheles albuminus, we use Anopheles darlingi, but our data are actually quite different. In other words, who infects the vector? Do you have to have visible parasites in your blood on a blood smear? Will people that are microscopy negative, PCR positive, will they infect? What happens over time? These are the big questions because, again, it's that area under the curve of malaria parasitemic days in relationship to mosquito density that really determines hypoendemicity. And so here's the data. We have about 60 patients that we've accumulated by now. And so these are three groups. They're half female, half male. We have patent, that means you see the parasites under a microscope, and these are symptomatic malaria patients. All of them, by definition, are blood smear positive. We have a patent but asymptomatic group, again, blood smear positive. Then we have a asymptomatic sub-patent group, and that's a logistical challenge, which I can talk about later. And what we find is that if you don't have visible parasites at that one time, you don't infect the mosquitoes. At least you do less than 1 in 23 times. So we think that it's extremely low. And we have we've done probably 1,000 membrane feeds in Iquitos over the years. We think that these are real data. We don't think that these zeros are artifacts. The other thing is the more parasites you have, it seems the more often you'll infect the mosquitoes in terms of oocysts. The more absolute parasites and more gametocytes you have, probably related to the higher oocyst count. Okay, so I'm not showing all the detailed numbers. We think that's an important lesson. And we need to continue these kinds of experiments because we just haven't been able to show that smear negative PCR positives, in other words, the subpatents, infect this colony of mosquitoes. And so we don't know, again, what the malaria parasitemic days, that area under the curve in the field that's going to be done over time. We've only done single time points. And so we're actually approved now to do direct feeds and indirect feeds, and we can actually serially sample asymptomatics over time, and that's part of our upcoming sets of experiments. Okay. Finally, I wanted just to show a little bit of how the isomer platform is leading to some translational research. This is work that was led by, that is being led by Elizabeth Winsler. Some funding from MMV and also the infrastructure of the isomer is what makes this possible. Pamela Orjuela from UCSD and Saira Villa, Carlos Tong from Iquitos. So here's our new building. Actually, it's a LEED Platinum building in San Diego. And this is Iquitos, Peru. So this is we constantly go back and forth between these places. And I'll just skip that. We do experimental feeds of the mosquitoes with Vivax-infected humans. We have no problems finding them because they're within 5 kilometers of the lab. And we use them for these people for producing sporozoites and other purposes. This is our baby picture. This is fairly recent where we're able to see at day 5 and day 7 exoerythrocytic forms in HCO4 cells in our laboratory cell culture done in Iquitos, Peru. So we bring our patients in, we infect the mosquitoes, we can produce about 10 million sporozoites of Vivax a week. We dissect the mosquitoes, clean them up, and put them into cell culture with the cells, and we can see small forms and we can see large forms thanks to Stefan Kappa for the antibodies to this up in sporozoite 54 protein. And we can actually take those cultures and add drug to them that we think either will or won't have effects on the hypnozoites. We think that we don't know the small forms are hypnozoites, but you can look at day 3 through day 9, and you can see that atovaquone doesn't have much of an effect compared to a negative control on the large forms, or the small forms, but new drugs from drug screening through the Winsler lab can do so. So we actually think we have at least preliminary data that supports using a Vivax sporozoite culture method in the endemic site, all based upon the isomer infrastructure to be able to do some Vivax sporozoite biology. Okay, so that's the acknowledgements. And so tying it all together, what we're showing is how the epidemiology, the vector biology in terms of mosquito differentiation, genetics, and translational work all come together to try to understand how asymptomatic parasitemia drives hypoendemic malaria. And so this is our theme that we now need to take the data and see how to apply it towards novel interventions, whether based on new drugs, whether based on new vaccines, or vector-aimed strategies. Thank you very much for your attention.
Okay, I think we're running a little bit into lunch. Maybe we have one question, and then we can approach Joe later for anything else. Thank you.
Thank you. Based on the parasitemia of Plasmodium vivax, it seems pretty high, given the fact that the reticulocyte number is pretty low. Is that the population you studied? Do they have a high percentage of reticulocyte?
No, the parasitemia is routinely run between maybe 0.05% and 0.8%. Rarely does it go above 1.
That's pretty high for Plasmodium vivax, right? Given the fact that when you have 0.5% to 1% reticulocyte, when you have 0.8% parasitemia.
Well, I don't have I don't have those numbers for you. It doesn't usually run so high, but we see a range for sure. 0.01. We can, again, we detect by microscopy down to 1 parasite per 20 to 30 microliters. So, and you multiply it against white cells, and you get your numbers, your absolute parasite counts and whatnot. But it is what it is.
We should look at that particular group of population. They have a high number of reticulocytes. We got to look at that.
Okay.
Okay, thank you very much. I'd like to thank all the speakers in this second session. I hope you enjoy your lunch.